Isolation of one or multiple cell types from a heterogeneous population is an integral part of modern biological research and routine clinical diagnosis and treatment. Purification of specific cells is essential for basic cell biology research, cellular enumeration in specific pathologies, and cell-based regenerative therapies. The central principle of separating any cell type from a population is to utilize one or more properties that are unique to that cell type. The most widely used cell isolation and separation techniques can be broadly classified as based on adherence, morphology, density/size, and antibody binding. The high-precision single-cell isolation methods are usually based on one or more of these properties, while newer techniques incorporating microfluidics make use of some additional cellular characteristics. A cell isolation procedure can either be a positive selection or a negative selection. The former aims at isolating the target cell type from the entire population, usually with specific antibodies, while the latter strategy involves the depletion of all cell types of the population, resulting in only the target cells remaining. Both types of isolation methods have their advantages and disadvantages. Due to the use of specific antibodies targeting a particular cell type, positive selection yields a higher purity of the desired population. On the other hand, it is more complicated to design an antibody cocktail to deplete all the non-target cells, making negative selection less efficient vis-à-vis purity. Furthermore, a cell population isolated through positive selection can be sequentially purified through several cycles of the procedure, a benefit that negative selective cannot provide. However, positively selected cells carry antibodies and other labeling agents that may interfere with downstream culture and assays. If that is a concern, it is preferable to use a negative selection method. To isolate a particular cell type from a heterogeneous population depends on the unique properties of that cell type. Surface charge and adhesion. This feature determines the extent of attachment of cells to plastic and other polymer surfaces and can be used to separate adherent cells from suspension/free-floating cells. Cell size and density. The physical properties of size and density are commonly used for the bulk recovery of cells, either by sedimentation, filtration, or density gradient centrifugation. Cell morphology and physiology. Different cell types can be distinguished by shape, histological staining, media selective growth, redox potential, and other visual and behavioral properties which can then be harnessed to isolate those cells. Surface markers. Specific binding of surface antigens to either antibodies or aptamers can selectively capture cells of the specific surface phenotype. The captured cells are subsequently detected with the help of measurable probes, usually fluorochromes and magnetic particles, with which the antibodies/aptamers are labeled. In addition, two or more of the above principles can be combined to increase the specificity of isolated cells further. Usually, such compound techniques consist of a label-free, the first three in the list, method along with a label-incorporating method. Adhesion properties of cells. Based on whether cells need to attach to a solid surface for survival and growth, they can be classified as follows. Adherent cells. These cells require a suitable surface to attach to thrive, for example, macrophages, fibroblasts, mesenchymal stem cells, etc. This attachment occurs through the interaction between the cell adhesion molecules, CAMs, on the cell surface and the corresponding ligands on the culture surface. Suspension cells. These cells naturally do not require an attachment surface and occur in suspension in the body, usually in fluids like blood and lymph. Examples include lymphocytes, granulocytes, and other immune cells. Cancer cells that have lost contact inhibition and anchorage dependency. In serum-free conditions without any anchorage facilities, cancer cells can form spheroids and amorphous aggregates. Cell isolation through adhesion properties. Cell culture systems are broadly classified as adherent and suspension depending on the anchorage dependency of the cells. Isolation of both adherent and free-floating cells can be tailored to the specific cell type by adding the requisite growth factors and suitable cell culture plates. Adherent cultures, example, macrophages, fibroblasts, mesenchymal cells. Macrophages are routinely isolated from bone marrow or peripheral blood. Right after the isolation of mononuclear cells, they can be seated on coated polystyrene plates along with serum and monocyte/macrophage differentiation cytokine cocktail. After 5 to 7 days, the cells differentiate and form an adherent monolayer while the unwanted cells remain in suspension and are discarded. Suspension cultures, examples, stem cells, embryoid bodies, tumor spheres. Cells naturally growing in suspension and cells that have lost anchorage dependency can be separated from the adherent cells by culturing in ultra-low attachment plates in the absence of serum. The desired cells either grow in a single-cell suspension or aggregate to form floating spheroids. The adherent cells die out without the support of an attachment surface. The advantages and limitations of adhesion-based isolation. Adhesion-based methods are technically simple, reproducible and mostly cost-effective, which yield a good crop of the cells. The limitation is that the purity of the recovered cells is low and there is always a risk of cross-contamination with other cells as well as with bacteria. Cell density/size-based separation. Separation techniques relying on the size and density of cells are frequently used to separate specific cell types from bulk peripheral blood and bone marrow. The most commonly used methods in this category include density gradient centrifugation and filtration. They are also called bulk sorting methods since they help isolate relatively large cell populations in a short duration. Although bulk sorting methods are an essential part of several clinical and biotechnology procedures because of high yields, the purity and homogeneity of the harvested cells are quite low compared to other cell separation procedures. The most frequently used density-based separation method is gradient centrifugation. The biological sample is centrifuged in a suitable gradient medium at the appropriate speed till the different cell types are fractionated into different layers or phases depending on their respective densities. Density gradient centrifugation is frequently used as an initial step to enrich for certain cell populations before more sophisticated isolation procedures such as FACS, MAX or single-cell sorting as well as organelle and nucleic acid extraction. Filtration-based cell separation is a size-based method wherein cells smaller than the pore size of the specific filtration device pass through and larger cells are trapped. Methods based on cell physiology and morphology. A specific cell type can be selected over unwanted cell populations by culturing them in a medium that provides some selective advantage to the desired cell type. In routine cell culture, a particular cell type can be enriched by adding specific growth factors and cytokines. Typical examples include enriching populations of a specific lineage and differentiation stage. The use of selective media allows specific cells to grow and inhibit others through antibiotics or specific growth inhibitors. Selective media is often used for isolating transfected cells and hybridomas. There are basically two approaches to use a selective growth medium, based on the type of selectable marker. Metabolic/biosynthetic enzymes. This strategy can be best explained by the HAT medium selection often used in hybridoma technology. HAT stands for hypoxanthine aminopterin thymidine. Aminopterin blocks de novo DNA synthesis and hypoxanthine and thymidine provide the raw materials for the alternative salvage pathway. The key enzyme needed for the salvage pathway is HGPRT, hypoxanthine guanine phosphoribosyl transferase, and only cells expressing the HGPRT gene can survive in the HAT medium. Antibiotic resistance. If the transfected cells express an antibiotic resistance gene, the specific antibiotic is added to the medium at a concentration that is lethal to the cells which do not express the resistance gene. Common antibiotics used for mammalian cell selection include bleomycin, poromycin and hygromycin. Growth selection of specific clones is routine in most cell and molecular biology labs and offers the following advantages. 1. Relative ease of performance. 2. Reproducibility. 3. Adequate cell yield. The limitations of selective media-based isolation are as follows. 1. The risk of contamination. 2. The emergence of spontaneous resistant clones that do not carry the gene of interest. 3. The high costs of some reagents and specialized media. 4. The time and labor required for the expansion and maintenance of the relevant clones. Laser dissection technology, first described in the early 20th century, basically involves cleaving areas or cells of interest from tissue sections using a narrow beam laser. A more refined, microscope-based form of laser dissection technique, laser capture microdissection, LCM, was later developed to isolate pure cell populations from heterogeneous tissues on the basis of their natural morphology or specific histological/immunological staining. LCM has enabled researchers to separate tumor from healthy tissues, stem cells from stroma and epithelial cells from parenchyma with high precision. Coupled with high-precision surgery techniques, LCM can even be used to isolate single cells. LCM has the obvious advantages of being high throughput, precise and adaptable to different tissue sources and applications. 1. Depending on the laser voltage and tissue structure, as many as several thousands of cells can be collected in a short period. 2. Due to the high-precision laser beams, the damage to adjacent tissues is minimal, and one tissue section can be probed several times. 3. LCM microscope and platforms are easy to operate and can be easily synergized with other assays. 4. A wide range of tissues can be subjected to LCM including regular hematoxylin and eosin stained slides, archival sections and immune-stained frozen/fresh tissues among others. 5. The lasers used are essentially low-powered and therefore do not destroy the structure and integrity of macromolecules. LCM collected cells can be successfully used for downstream assays requiring functionally active nucleic acids and proteins. However, LCM is not without limitations. 1. For removal of selected cells post-laser targeting, the tissue sections cannot be coverslipped. Without mounting medium and coverslip, the refractive index of the tissue is altered, and that makes it difficult to visualize the desired cells. This problem is of particular concern in tissues with amorphous structures, such as tumors and lymphoid tissues. 2. Most LCM platforms employ lasers whose minimal spot size is 7.5 micrometers, and this is not precise enough to isolate single cells. 3. Another technical challenge that results due to tissue drying, especially frozen tissues, is the lack of adherence of the dissected tissue onto the film or inverted cap. Cell surface marker-based techniques. Cellular surfaces are populated with a multitude of ligands, proteins, carbohydrates, and glycoproteins, which perform various biological functions and give every cell a unique surface phenotype. Many ligands are antigenic and elicit an immune response in an allogeneic body. Therefore, specific antibodies raised against cell surface antigens can be used to target the cells expressing those antigens. Antibody-mediated cell detection and isolation techniques became commonplace with the discovery of the cluster of differentiation, CD, markers, surface receptors usually involved in cell signaling and adhesion. In addition to antibodies, surface molecules can also be detected by aptamers, single-stranded oligonucleotides that can bind to the several different targets simultaneously and detected by fluorescent or magnetic probes like antibodies. Immunopanning is another technique that uses specific antibodies coated on plastic petri dishes to enable the adhesion of cells with cognate antigens. The fluorescence-activated cell sorter, FACS, was invented by Bonner and Herzenberg for sorting viable cells with the help of fluorescent probes. The population stained with different fluorophore-tagged antibodies can be separated by the different fluorescent signals that they generate, in terms of wavelength and intensities. FACS is used exclusively for the positive selection and isolation of cells. Flow cytometry-based sorting of cells is now a standard procedure in many research and clinical laboratories. With the ever-increasing sophistication of FACS instruments and analysis software, more and more applications are turning to FACS for analyzing as well as isolating various cell types. 1. FACS is a highly sensitive and high-throughput procedure for isolating cells from heterogeneous populations. 2. It is the ideal method of simultaneously sorting multiple populations based on just their immunophenotype. 3. FACS is a highly versatile technology that can separate cells based not only on surface markers but also cell size and granularity, cell cycle status, intracellular cytokine expression, metabolic status, etc. The technology of FACS has the following limitations and challenges. 1. Although FACS can help sort several populations simultaneously, the sorting process itself is slow. 2. The recovery of most FACS sorters is around 50% to 70%, making it necessary to begin the sort with a high number of cells. This requirement poses a challenge when rare cells like stem cells and CTCs need to be sorted. 3. As the sophistication and precision of FACS instruments increase, so does the probability of technical errors. The operation and maintenance of FACS sorters are expensive and also requires highly skilled personnel for handling, troubleshooting, and repairs. 4. Since FACS requires single-cell suspension, information regarding the tissue architecture and intercellular interactions are not available. 5. Even though advanced FACS sorters provide the option of detecting as many as 10 to 12 colors simultaneously, it raises the problem of spillover of the different fluorophores into nonspecific channels. The spillover occurs due to the overlapping spectra of the fluorophores and lowers the resolution between populations that have closely related immune phenotypes. Immunomagnetic separation of cells is based on the deflection of cells in a magnetic gradient field. Based on the type of magnetic particles used, immunomagnetic separation are mainly of two kinds, Dynabeads-based and MAX technologies. Immunomagnetic techniques can be used for both positive and negative selection of cells. Dynabeads are hollow, spherical superparamagnetic polymer beads available in sizes ranging from 1 to 3 micrometers. These beads exhibit magnetic properties only in a magnetic field and have no residual magnetism once the latter is removed. Dynabeads can be coupled with antibodies or other ligands to targeting of specific cells or immunoprecipitation reactions. The magnetically activated cell sorting technology was first developed by Miltenyi Biotech in the 1990s and since then has become a staple in cell culture laboratories and clinics for the rapid and bulk isolation of desired cells. The target, or non-target cells depending on the selection strategy, are labeled with 50 nanometers magnetic microbeds conjugated antibodies and then subjected to a magnetic field. Advantages. 1. MAX is a high-throughput, selective and rapid method for isolating target populations or removing undesired cells. 2. The MAX columns help isolate rare cells with high specificity. 3. MAX can be scaled up or down depending on the desired yield and the downstream applications. 4. It is a highly versatile technique and can be extended to a wide range of cells and over various platforms including microfluidic devices. Disadvantages. 1. In case sorting with dynabeads, the latter need to be eluted out owing to their large sizes as it may affect downstream assays. 2. Separating cells using magnet separators is not very efficient as draining out the unbound cells still leaves many unbound cells in the tube along with the bound cells. To improve the purity of the sample, the sorting step may need to be repeated several times. 3. For delicate cells, binding of the magnetic particles may cause mechanical shear. MAX columns should be used instead of separators to minimize the shear. Aptamers are single-stranded oligonucleotides, DNA or RNA- that can bind to highly specific targets based on structural conformation. Aptamers were first generated using a procedure called CELX, systemic evolution of ligands by exponential enrichment, which involves the sequential binding of the oligonucleotides to target molecules. The targets of aptamers have evolved from simple ligands like surface proteins. The first DNA aptamer was designed for the human thrombin, to more complex ones like RBC membranes and even whole cells. Aptamer-based cell detection. Once aptamers are selected for whole cells or specific biomarkers, they can be conjugated with different biosensors like fluorophore, luminophores, nanoparticles, etc. for rapid detection of target cells. Depending on the signal output, the aptamer-based assays are classified into 4 types. Direct binding. It is the simplest assay where aptamers labeled with a single moiety of a fluorophore or a luminophore directly bind to the target and the signal can be easily read out with a suitable detector. Target-induced structural switch. In the absence of the target, the fluorophore-labeled DNA aptamer forms a partial duplex with another, nonspecific, aptamer labeled with a quencher. When the target is introduced, the aptamer binds to the target, thereby releasing the quencher from the fluorophore and triggering the latter's signal. Sandwich binding. The specific unlabeled aptamers immobilized on a solid phase first capture the target cells, which is followed by the addition of the biotinylated version of the same aptamers. The signal is generated through further binding of streptavid and conjugated HRP to the biotin molecules as in ELISA. Target-induced dissociation. DNA aptamers coiled with gold nanoparticles, OMPs, are used for this assay. As soon as the aptamers see the target, they uncoil and bind to the target cells and release the OMPs. The latter are precipitated in a salt solution bringing about a color change. Aptamers offer several advantages, especially over the antibodies-based isolation methods. 1. The preparation of aptamers is completely in vitro and thus obviates the use of live animals and highly sensitive hybridoma cultures that are required for the generation of monoclonal antibodies. 2. The repertoire of specific aptamers that can be synthesized for targeting any cell is virtually limitless. 3. The chemical synthesis of aptamers and the scale-out PCRs are easy to perform and cost-effective. 4. The aptamer assays are highly reproducible, and variability is generally very low between different batches. There are some potential limitations as well. 1. Due to the vast number of aptamers generated, there is always the risk of nonspecific binding. Therefore, several clones need to be tested for specificity, which can be time-consuming. 2. The cell yield is very low, which does not make it an ideal method for the isolation of rare cells. 3. The safety and biocompatibility concerns of aptamers have not been addressed so far, therefore it not yet approved for clinical applications. Certain cell isolation techniques make use of more than one cell characteristic, as classified above, and are therefore the combination of at least 2 different techniques. The objective is to either combine the advantages of both techniques or overcome the limitations of one of them. The combination is usually that of an immuno-label-based technique with a label-free technique utilizing a cell property like density, size, morphology, etc. Thanks for listening. If you have any questions about cell culture, please feel free to contact us in the following ways.